So here is another video of the processing of the mRNA. So in prokaryotes, the mRNA are rarely processed, but in the case of the eukaryotes, there is a regular phenomenon. So through removal of the introns and the addition of the exons causes the modification of the mRNA, forming the pre-mRNA to the mRNA by a process known as splicing. So the two boundaries between the two exons and the introns lying between them are known as the splicing junctions. And the primary RNA transcript of a structural gene is called the pre-mRNA, from which the mRNA is generated by splicing. And the pre-mRNA are collectively called as heterogeneous nuclear RNA, that is hnRNA, because of the large variation in their sizes. The hnRNA exists in the form of ribonucleoprotein RNP, particularly, and is called as the hnRNP. And hnRNP particles takes the form of a series of beads connected by a fiber. A total of 20 proteins are associated with the hnRNA of these core proteins like A1, A2, B1, B2, and C1, C2. So it can be cleared by the through a figure. So processing of pre-mRNA occurs at two steps, that is the 5' modification and the 3' modification. So first we are saying we are going to see the 5' modification, that is 5' capping of the pre-mRNA. With the help of the RNA triphosphatase, guanylyl transferase, and guanine 7-methyl transferase enzyme, the addition of the 7-methyl guanine at the 5' end and called as the 5' capping of the pre-mRNA. So next is the 3' polyadenylation. So in this sequence, the 3' end, the addition of the polyadenylated A. So through a series of steps, it can be seen like there is a sequence of in the mRNA like a poly A signal and the GU rich element, and in between them there is a polyadenylation sites. So binding of CPSF, CSTF, and CF, the CF1, CF2, and CST5, known as the cleavage stimulation factor, cleavage and polyadenylation specificity factor, it eventually forms the polyadenylated mRNA, that is AAAN. So 5' capping of pre-mRNA and the polyadenylated mRNA occurs in this process. So in this figure, there is an exon and intron side, and exons are added by the splicing of the introns, and there is a steps like first and second transesterification of the reaction and forms the splicing of intron in pre-mRNA and form the mature mRNA. So we can say that the processing of mRNA can be done by 5' capping of the pre-mRNA, polyadenylated mRNA at the 3' end, and the splicing of introns. So all about the processing of mRNA. Keep watching. Thank you.